CIEHS, or Center for Integrative Environmental Health Sciences, TRSC seminar series. So this is a core within the CIEHS, and so we're doing quarterly seminar series now about some of the services that we offered, and this will be I think the second seminar in this series in this kind of new format anyway. Jiapeng gave one on clinical research services, and so it's a relatively new thing. And so for today's talk, there'll be several presenters. So I'll start off, and then actually being back up, Luis, Dan, Conklin, and Ken Palmer, and then Jiapeng Huang will talk as well. The first three will be about some of the basic science research that we can support through this core. And then Jiapeng is going to talk again about clinical research, because since the last time we've had one of these seminar series, the notice of award for the CTR-D came, and I think hopefully all of you are aware of that, and so that can impact maybe some of the environmental health clinical research that we do as well at UofL. All right, so next slide. So 4 presenters, we've got 35 slides. I think we'll make it through in time with plenty of time for questions and answers. So I'm going to lead off with a little bit of overview for CIEHS and the TRSC. So back up one, Luis. I'll stay on that one. Thanks. And because I did notice that there are few people on the call that are maybe not as familiar with the center or the core, so I'm going to give a brief overview of that, and then I'm going to talk about some of the basic science research support services. So I'm going to go over all the different types of research support services that the core can help support, but today we're going to focus on the basic ones. And as a component of that, some of the types of things that we can support will be animal phenotyping and histology. And then Dr. Conklin will talk about some of the inhalation exposure facilities and other facilities available. And Dr. Palmer will talk about some of the RBL type facilities and pathogen exposure. And as we go along, I'll through the introduction talk about how all these things can be linked together to support a project. And then Jiapeng will finish up circling back around to clinical research and CTR-D. So next slide. Okay, so Center for Integrative Environmental Health Sciences is a NIEHS funded P30 mechanism center. And so we're had just recently, over the last several months, April, I believe, submitted our first competitive renewal application. And so what the objective of the center is to study chronic diseases. And you can see this diagram with this wheel. And so we look at kind of interactions between exposure to pollutants, on the right, and then going around clockwise, disease modifiers such as life stage, genetics, sex, and then things in the social domain as well. So smoking, diet, alcohol, socioeconomic status, etc. And we are trying to be consistent and forward thinking, consistent with the new strategic plan for environmental health research from the funding agency, the NIEHS, and looking at things related to something called exposome, precision environmental health, environmental justice, health disparities, climate change, and then mechanistic toxicology. And so I think this is a great figure that Dr. States and others have developed. Dr. States is, of course, our leader and center director. And so one of the interesting things that we deal with in Kentucky is what Chris calls this urban-rural environmental exposure paradox, where we have some areas in Kentucky out in the, and I'm from Kentucky, I'm a native, so I could say this, places out in the middle of nowhere, out in the middle of farm country that might have chemical plants. and significant exposures that you might expect from an urban environment, and then vice versa with rural type exposure, such as, cornfields that we might see with Roundup on them just driving around Louisville. And so the center has a very strong translational research vision, and TRSC is the core that supports the translational research vision of the center. All right, so next slide. And so the specific aims of the competitive renewal are shown here for this core. So we want to recruit and engage investigators to translational environmental health research. And this is one of the reasons why we have having this talk today, is to get people to learn about translational environmental health research, and hopefully get them interested and motivated in doing that. That could be existing basic science researchers that want to extend their research into human subjects, or even clinicians that had a clinical observation that might want help developing an animal model and in need of a team to help do that. And so we want to help support existing extramurally funded environmental health science. That's what EHS means, stands for research project. And then we also want to help develop new interdisciplinary center research. And so one of the things we do is provide intellectual support through consultations and help investigators to build teams to take a team science approach, because you really can't do translational research with without a team. And then one popular and I think successful way we've been able to achieve some of these objectives is through the core utilization subsidies, which have recruited many members into the center. We used to call those research vouchers, but we're changing the name to core utilization subsidies to better reflect what they're for. So the CIEHS is a core facility center, and so it's composed of cores, and it's an interesting type of NIH application because it doesn't propose specific research project. It provides cores to support research, and it also funds pilots and can financially subsidize utilization of the cores. And then the third aim is to provide access and the services and resources that foster integrated and translational health research. And that's really what this talk is about today, some of the services that are offered, just to make sure that people are aware of them. Okay, next slide. And so when we define translational research, what that means, we use something called the NIEHS Translational Research Framework, which is published and available online. And based on the needs of our members, we've modified the translational research framework, or adapted it for purposes of to explain this core and define what we're really going to focus on supporting. And so we kind of really focusing right now on several domains of research: basic science, clinical science, GIS, and population health and epidemiology studies. And so what we're really defining translational research as a project that's really crossing over these domains, so involving more than one of these domains. There's certainly other definitions of translational research, but for the purposes of our center, we ultimately want to see research cross several of these domains. Now, it doesn't mean that where you're at now on a project, maybe you're starting a new type of exposure in a mouse and you ultimately want to go into humans, but you're not doing humans now. That still, would be translatable in the future. And so the core offers support for each of these areas: basic science, clinical science, GIS, epidemiology, and population health. And we've assigned a navigator to each of those areas. So I'm the navigator for basic sciences, Jiapeng is for clinical science, Natalie Dupre for population health, and then Charlie Zhang for GIS. And so we're having these seminars with each of the leaders from each of these different rings, so to speak, to give a little overview of some of the services that their core offers. And it ends up being right now that this core is mostly kind of a, virtual or kind of flow through kind of core. So we connect people to existing university resources and help work with the existing resources to leverage them for environmental health science, and then link these rings together to promote translational research that ultimately is going to move across these. And so Jiapeng gave a talk in April, I believe, about the clinical sciences, including resources for research, including the clinical trials unit. And so today we'll be focusing on most of this talk on the basic sciences, and then Jiapeng will give us a brief update on this clinical sciences in the context of the new CTR-D. So you can see the navigators here for each of those. All right, next slide. And so what are the basic science resources? So me and Dan and Ken, we're going to talk about some of these, but briefly, some of them are listed here. Study design, we can provide some help with IACUC, and then, importantly, Amanda LeBlanc has had a leadership role with IACUC for years. She's a center member and works with the core, so we can help with those issues, or at least support them. We're not going to write your IACUC and get it approved for you, but we can help you along the way. And then we're interested in environmental health science about exposures. And so there's many projects are involving mice. This is, again, basic. So most of our support is for mouse studies or rodent studies, but there's certainly other things that we can do. But we got to expose these mice somehow to environmental pollutants. And then Dan and Ken have really been teaching me, how to broaden this vision and include different types of exposures, such as to pathogenic agents. So they'll talk about that a little bit as we go along. And so we expose the mice, and then we can look for effects of the exposures, and that includes behavioral and metabolic phenotyping. And then we can also help with histology and pathology services. So I'm not going to go through this table. This was submitted with our grant, but basically members that have accessed the core have been very prolific and productive and have achieved a high grant funding success rate. All right, so next slide. All right, so I'm going to delve in here, kind of a little bit of a transition point, and talk about some of the specific basic science research services. I think I've got one or two slides here, and then we'll flip over to the different presenters. And so again, this is primarily rodent studies, and this is based on the needs of our members. But then if you drop down to the last bullet point, we can provide linkage to other members within the center that are doing a lot of other types of animal research. So there's obviously other models like zebrafish that are being used at UofL, but then there's some really neat wildlife sentinel type of animals that are being studied too. So these would be like marine animals, such as the WISE Lab studies. We also have investigators studying songbirds, and so these can be leveraged for specific types of studies. And I can help connect you with those laboratories and their leaders if people are interested in that. But I think Dan's got some talk about the mobile animal exposure unit, or at least mention that. And then the center is developing some rodent imaging. So right now we're not necessarily supporting or imaging for animal studies of environmental health, but they are being developed, and that's something we may add in the future. Next slide. And so when I say this is kind of a virtual core. So some of these connections will be through other cores that can actually perform the work, or centers, as the case may be. So I also serve as a director of the Animal Model and Biorepository Core through the COBRE Hepatobiology and Toxicology Center. And so through that core, we offer several services. And I saw Rand was on the call, and we met with him yesterday. So this COBRE is going into phase 3. We're working on the renewal application now. And so we really kind of want to open up access to this core, and so that it can become more self-sustaining, and operate on a fee-for-service type basis. And so we do several things in the core. We do have some biorepository services that are focused primarily on liver disease. But for purposes of this talk today, I want to talk about some of the animal phenotyping services. And so this core is over in the CTR building. And so would be really, great for people here, but can also be accessed for people in other places. Animal phenotyping is incredibly important for people interested in really any disease, but particular metabolic diseases. We have a MRI here, echo MRI, for body composition analysis. The DEXA scans are available elsewhere, and they're a very good tool. The MRI is nice in that it can be done in live animals very quickly without anesthesia. And so it gives body fat, muscle, that type of thing. And we also have some great equipment for making slides for histology. And a lot of this is automated, and there's shown on the lower right a automated processing unit. We even have an automated stainer here. So right now it's kind of working as a we train you to use the equipment, or your technicians, and but in the future we hope to make it a more full service model, where you drop off your cell blocks or whatever, and we give you slides back in exchange for some money. So Rand, if you're still on, I'll be working with you more on that. But we're gonna, try to encourage utilization of these types of resources by environmental health researchers beyond the liver center and into the broader environmental health center. So next slide. All right, so I'm going to go over to Dan here, who's going to talk about some additional services.
Yeah, thanks, Matt. And as Matt pointed out, the goal here is to make everyone aware of the services that we have available on the HSC, and also to improve chances of getting funded through collaborative work. And so this is a busy slide, but 10,000-foot view, the kinds of things that our core is involved with are the direct exposures of animals to things like particulate matter, volatile organic compounds, tobacco products, and biogenic VOCs. We monitor all of these exposures, whether it's total suspended particulates or fractions of the particulate like PM2.5, or the VOCs through photoionization and EC detection with these handheld detectors. We also collect biomarkers of exposure. So this is important if you want to do pharmacokinetics. After a specific exposure, we can collect blood and urine in time-sensitive ways. Phenotyping, as Matt mentioned, this is obviously very important depending on your outcome. We do a number of phenotypic measures during the exposures. So these are real time using telemetry. And our system is the Data Science International, but we can do electrocardiogram, blood pressure, blood glucose, EEGs and EMGs, temperature and activity. So these can be very important for collecting data during acute exposures when you really want to see changes on a real-time basis. But of course, this can be done post-exposure. And as Matt pointed out, sometimes the equipment appropriate for a certain measurement are not going to be in the exposure facility or right where you may be working with your animals. So there may be some logistical hurdles that we have to get over. And as Matt mentioned, we'll help you write those protocols for IACUC so that animals can move from, say, an exposure to a location where they can be phenotyped. But some of the post-exposure measures we do include DEXA scans, so that would be equivalent to the MRI. Echo, this is done with anesthesia, but we have that system set up, and the animals recover from this. So it's a 5-minute scan. So that's really easy. These are all these tolerance tests that we do: glucose, insulin, pyruvate. We also have a Promethean system for food and water intake, activity, VO2, VCO2, etc. We also do a number of ex vivo phenotyping, certainly targeted towards more cardiopulmonary, but there's opportunities for tox screen, CBCs, flow cytometry for specific markers using cytomics. Of course, these are in collaboration with other groups, and I just want to make you aware that this is a kind of a holistic approach. Next slide, please. Just keep, there you go. So who's the team that currently is working in our core? Show them here. We have a group of 5 individuals plus me that are either full-time staff, where we have Sarah Barnett just came on hand to do some of the weekend exposures in our facility. But we are a full service, and I'll talk a little bit more about the inhalation facility as a location in a second. But it's received, as Matt mentioned, it's been important in receiving a variety of funding over the years, including some ongoing funding with our Superfund Center run by Sanjay Srivastava, but also the current funding I have that takes advantage of the inhalation facility for a variety of exposure conditions. Next slide, please. So some of you may not be, okay, before I talk about the location, also our staff, even though we offer all these services, we also offer the training and mentoring for use of the equipment. And so that's kind of important. Maybe you haven't, you know where the place is, but how do you get to use it? And can your students use it? Can your staff use it? Can you use it? And the answer is yes. And we provide training for everyone, and you become certified then. Certainly if it involves animals, you have to have an IACUC and so on and so forth. Again, we'll help you with that. Next slide, please. But where is the inhalation facility? It's a suite that was put on the 7th floor of the MDR. Back in 2008, we received support from the EPA to establish a particulate matter center, and we also received funding over $1 million to renovate this to be a barrier access facility, so we could house animals in there and then perform exposures. So this is an example of the rooftop where we collect ambient Louisville air and bring it down into our system, shown here as a VASIS. It's a versatile aerosol concentration enrichment system. And that takes that PM 2.5, a very specific fraction, and concentrates it about 6 to 10 times, and we call that concentrated ambient particles, or CAP. And this is just a demonstration of a filter and how dirty, it gets akin to your air filter at home through your furnace, and you know that it's time to change it when you see it turn gray. Next slide. Okay, here's some of the rooms in there. The suite proper is called MDR 722, and that has card access only into that room, and it's attached with an animal room. But we have several other rooms that are dedicated to, we have a surgery suite for doing implanting the telemeters or osmotic mini pumps. That's over here. We have a tobacco product exposure room dedicated to e-cigarettes and conventional tobacco cigarettes. And all of these rooms have been outfitted. You see the red film over the doors. All of these have been outfitted with that so that we can convert them to perform exposures in the dark, or at least under red light, if you were interested in circadian rhythm. For our VOCs, we perform those in a fume hood, and here's a photoionization detector for detecting things like acrolein and benzene and formaldehyde. And this is a Kintec oven that controls the aerosolization or vaporization of these materials. Next slide, please. Okay, some of the phenotyping that we discussed, including telemetry. This is a DSI system in which we can control or monitor heart rate, activity, blood pressure during the exposure and after in the home cages. We also measure blood pressure non-invasively through the tail cuff. This allows us to have a little bit more high throughput as each animal doesn't need to be instrumented for this, and you can measure it anytime as needed. And then we also collect urine. So we have a urine collection system. Here you see 8 chambers, individual mice, and we can do pharmacokinetic style collections on those. Other instrumentation in other facilities are the DEXA scan and our ultrasound. Next slide, please. So one example of a CIEHS funded pilot award was to look at the combined effect of hypertension. As Matt mentioned, from a translational standpoint, this is the most highest incidence cardiovascular disease in the world is hypertension. And so, but we know very little about the mechanisms by which hypertension will change the response to an exposure. And so this is an example of a mouse that got Ang2 induced hypertension and then was exposed to ambient air pollution. Here's a real daily trace of the exposure levels from last summer, that on the day when the wildfires reached our city, you can see the huge spike in the PM levels went up over 1,800 micrograms per meter cubed in our chamber during that particular day. But you can see it changes on a daily basis. So the funding for this was to look at the combined effects of hypertension and CAP, and I can happily say that the preliminary data from this study, which included blood pressure and ex vivo measurements of aortic reactivity, led to funding of a DoD grant recently. So that was good return on investment on that and use of the inhalation facility. Next slide. And the last one, which is a nice segue into Dr. Palmer's presentation, is that we've been collaborating on combining exposures of animals in the inhalation facility, also with and without hypertension model, and then press the return one more time. Oh, go forward. There we go. Thank you. Where we implanted mice with osmotic mini pumps. They stayed in the inhalation facility, received an exposure to a VOC, and then were transferred to the RBL, where Dr. Palmer and his team infected them with SARS-CoV-2, and then tissue harvests occurred at 10 days later. So this represents a real collaboration, also a funded pilot award from the CIEHS that is trying to address in isolation a single exposure. Obviously, the exposome is much more complicated, but how to disentangle the real world is to basically collaborate across these disciplines to better understand how those exposures may increase the susceptibility. And we add in the physiological or pathophysiological condition of hypertension, which really does represent really some major aspects of what people face in the real world. And with that, I'm going to turn it over to Dr. Palmer.
Thanks very much, Dan. Happy to follow that. As Dan and Matt had mentioned, we are collaborating with CIEHS the Center for Predictive Medicine is collaborating with CIEHS to help address infectious disease exposure as one aspect of the exposome. And within the CPM, our major reason for being is to address emerging and re-emerging infectious diseases. And we all became all too familiar with emerging infectious diseases over the last few years. And I think it's going to be an issue which continues to challenge us and will affect the response to infectious disease with increasing issues with climate change and environmental exposures. So to introduce you all to the CPM and the Regional Biocontainment Lab, which is hidden away on the corner of the Shelby campus, we have a really impressive state-of-the-art research building in the Regional Biocontainment Lab on the Shelby campus, which is a large, around 50,000 square foot research lab that is essentially a shared research facility with biocontainment labs that allow us to do both in vivo and in vitro research on infectious diseases. The Regional Biocontainment Lab supports UofL and CPM faculty research programs. We have operated on a fee-for-service model where we provide services on behalf of for other people, other researchers, both within UofL as well as in the private sector and outside academic institutions. The Regional Biocontainment Lab employs 10 to 12 regular on-site professionals who are involved in facility operations, in operating the vivarium, in the biosafety operations, and in operating the shared research facilities. Next slide, please. Some of the agents that we have here available, we have a large portfolio library of infectious disease agents that people might be interested in working with. Some of them that I'd like to highlight, obviously multiple drug-resistant bacteria, Klebsiella, Pseudomonas, Acinetobacter. Sorry, I stumbled over that one. We also work with MERS coronavirus, SARS coronavirus 2, the original SARS coronavirus. We work with influenza, including emerging H5N1 strains, and a lot of mosquito-borne infectious diseases, chikungunya virus, Venezuelan equine encephalitis virus, West Nile virus, Zika virus, pathogens that are unfortunately going to become more and more problematic as we encounter climate change and changes in the distribution of insect vectors. Next slide, please. So we, in the vivarium facilities that we have at the Regional Biocontainment Lab, we're equipped for various different research animal models. The large majority of our work is done, of course, with murine models, but we also accommodate hamsters, including golden Syrian hamsters, and we've recently invested in developing a robust colony of Roborovsky dwarf hamsters, which we brought in because these animals are the same size as mice, approximately, and are very susceptible to SARS coronavirus 2, all of the emerging variants of concern, etc. We can accommodate ferrets, which has been a long-standing model for influenza, guinea pigs, cotton rats, and rabbits. So we have the ability to work on a number of different models, but as I say, mainly on mice. Next slide, please. We have benefited recently from a lot of new investment in biocontainment facilities from the federal government through G20 grants and a recent UC7 grant. And I would say we're exceptionally well equipped to do both in vivo imaging and as well as other cutting edge state-of-the-art animal research. We have facilities for doing live whole body imaging, including luminescent imaging using the IVAS system. We have new equipment to do CT and PET scanning in rodent models. We have equipment for assessing sensory perception, the startle response system. For in vitro research, we have equipment for doing cell sorting as well as doing flow cytometry. We have cytokine, high throughput cytokine analysis equipment, HemoVet for CBC sampling, and all the standard scientific lab equipment that you would expect to see in any research lab. Next slide. We have recently acquired new equipment for addressing emerging infectious diseases, and in partnership with Dan Conklin, who has helped us try to bridge infectious disease research with environmental exposure research. We have acquired telemetry systems. We've bought a Promethean metabolic containment caging. We have equipment for assessing lung function using whole body plethysmography. We have acquired equipment for aerosol delivery. We have a citation C10 confocal imaging plate reader, and as well as new and expanded flow cytometry. As I said, the Sable Systems Promethean core metabolic system we acquired strategically so that we would have equipment that is also present in labs that in the Center for Environmental Health Sciences and the Center for Cardiometabolic Sciences, so that we could bridge those two areas of research. And the Promethean system we're very excited about having and have started using it. It allows us to assess body mass activity monitoring in the animals, looking at respiratory output. And this system has been modified so that the caging system allows us to contain the infectious agents. So we can use it for pathogens that require both ABSL2 as well as ABSL3 containment. Next slide. We have the CIREC VivaFlow system for whole body plethysmography, which we've acquired with the equipment to accommodate 4 chambers. We have the equipment sized for using plethysmography to assess lung function in hamsters, and a separate system for looking at lung function in mice. Next slide. We purchased the CIREC N-Expose system, which allows nose-only aerosol delivery for up to 12 animals at a time. And Next slide. That finishes my presentation. So essentially, we've tried to set up a system to bridge environmental research with infectious disease research, and hope that we can serve our colleagues across the institution who are interested in environmental exposure and how various different environmental exposures might affect the pathogenesis of infectious disease. One of the areas that we're continuing to invest in is obviously in climate change, and we're acquiring equipment that will allow us to keep animals at temperatures that are more associated, unfortunately, with global heating. So I'll turn it over to Dr. Huang now, and we'll be happy to answer questions.
Thank you, Dr. Palmer. I think most of you know me. So my name is Jiapeng Huang. I'm an anesthesiologist. I also work with the CIEHS for the last 3 or 4 years. Really, CIEHS is really the place that made my research possible. I was a private practice physician before 2019. CIEHS took me on and funded me to do my initial research, and now my research career is taking off. So we're really excited. So on July 9th, we were notified with a notice award from NIGMS for the clinical and translational research development grants. So that's $11.5 million over 5 years. And then University of Louisville, VA Hospital, Norton Hospital, and Owensboro, then total committed another $11.5 million. So we totally have $23 million investment. I believe this is the largest investment in clinical research in the history of the university. So very excited. Dr. John Klein is the director. I serve as deputy director. Dr. Medicave is the core director for our health research. Dr. Palmer is the co-director for the professional development core. And Dr. Mayun Kong is our RDCD core director. So next slide, please. So what we will be able to do is really to establish a Louisville Clinical and Translational Research Center, LCTRC. So the purpose of this, really the vision of this, to build a high impact and durable clinical translational research program at the university. Our goal is to improve the health of Kentucky communities. This is not really only about Louisville, it's really about the rural Kentucky too. And that's why we chose Owensboro to work with. And if we do this well, and if we together do this well, I think the impact can be significant. We will be able to build and grow an integrated clinical translational research program. Really, our goal is to make this CTR a single point of entry for researchers. So if they want to do clinical translational research, you'll come to Louisville Clinical Translational Research Center, and we will guide you through this complex process. I think a lot of physicians are very scared, are very afraid of doing research just because it's so complex, heavily regulated. And as physicians, we don't understand the molecular pathways much at all. So for us to really learn all those, it's very difficult to do. So hopefully this center will be the single point of entry. And we want to develop and encourage community participation in all stage clinical and translational research. We want to address the issues that are important for the community, not only for the researchers. And the other thing is, I think the other frustration as a physician to do research is we don't have the personnel. I mean, I work, 100 hours per week clinically. I just can't do all the research by myself. So I need people to help. I do not have the physical infrastructure, and I really like what Matt and Kenneth and Dan put together. And there are a lot of resources we just don't know. So hopefully with the center, we should be able to guide whoever wants to do clinical research through the process. Next slide. So our partners at University of Louisville Health, as you all know, there was no UofL Health before 2019. So after 2019, UofL Health took over multiple hospitals. So right now we have 11 hospitals, 4 medical centers. We have 13,000 team members, and we have over 1,000 physicians providers. So it's a huge system right now, and it's doing really well, which is great. But what that also creates a really challenge for doing research. because our research infrastructure is not used to this kind of patient volume. And so that's what they need for the center. We work with Owensboro Health. We actually reach out to them multiple times, and they actually really don't have much support from their system. There's only one person to do clinical research for Owensboro Health, and however, they can be part of the research clinical trials, and it can do a lot of community engagement research. Next slide, please. And so we believe the center is transformative. I think this is the word we like to use, and innovative. And so I think the key point we try to do is we try to make it simple. We try to make it as a single point access to expand clinical research capacity at UofL and the affiliate health system. We want to provide this as a hub for the cores at the center and also the UofL cores. Such as the COBRA, the INBRE, the CIEHS, and all the other facilities that we have. We will create a Center for Clinical Research Innovation to tackle a lot of new technologies, right? AI, machine learning, how to use those in clinical research. And we want to make sure we have faculty committed to doing clinical research. And we want to strengthen the collaborations. And we want to expand our outreach to other CTR awardees across the nation. Next slide. And so this is the Louisville Clinical Translational Research Center organization chart. So this center is strongly supported by the UofL top leadership, from the President Shazel, the Provost, and also the Executive Vice President for Research Innovation, who happens to be the PI for the project, Dr. John Klein. So these are our healthcare partners: UofL Health, Owensboro, Norton Healthcare, and VA. So we are putting together external advisory committee and also steering committee. I serve as the deputy director. And the other thing we're pretty proud is we actually create a financial management center. So Dr. Kim is the director. For grant this size with the sponsor money and also the university money, it's very difficult to navigate through. So we create this center really to, for us to learn how to deal with this kind of complex funding system. And this will be useful for other members too. We hope that many people will get a grant in 10 million, 20 million rand, then we can potentially help from the financial side. Dr. Cunningham will serve as a COO. That's also pretty new. We have a chief operating officer. And John and myself will lead the core for administrative core. Dr. Keeve and Dr. McLean will lead the health research core. Dr. Palmer, Dr. Boyaki will lead the professional development core. Dr. Cohn will lead the research design, compliance, and data core. And then we'll also have the community engagement core. Dr. Dunn and Dr. Keita will lead that core. Next slide. I will kind of go over some of the cores and really to share some of the resources that will be available pretty soon. So the grant started, we got the notice we're on the 9th, but we can reach the date to July 1. So the grant is started this month. So for the health resource core, where Dr. Keeve is the co-director, we will select new pilot projects. So those are the projects they want test ideas with the potential to develop an independent application, key award or award. And we also want to support developmental projects for 2 years. And those are substantial amount of money too. For the pilot project, we're thinking $75,000 per year. For the developmental project, we're thinking $125,000 for 2 years. Really, the developmental projects really focus on the PI, and we try to make sure we want to help them to become independent researchers. And we will pair those project leads with physician scientists who are experienced with clinical trials, and we will monitor their progress. Next slide. And the other core is very exciting, is a professional development core, where Dr. Kenneth Palmer is the director, the co-director. And this has really got everybody excited. They actually presented a lot of new pipelines for the future. And we all know right now we're losing physician scientists, we're losing scientists doing clinical translational research. So we try to build a pipeline. One of the things we want to do is the UofL presidential scholars. So I'm going to show you on the next slide there's a well thought out, well developed plan to help early stage physician investigators and for them to secure grant funding and how they can give back, serving as a mentor for future. And this core will build interdisciplinary teams and networks. And there are two programs that are very exciting that actually Dr. Palmer and Boyaki are co-leaders for the physician scientist program at UofL, focused on the MD PhD students. Actually, this summer we have MD PhD pipeline students. I'm very lucky to have one student in my lab, and this is, those are the people that has a great potential to become physician scientists. Then the Dean Scholar program, I'll talk about just a little bit. And then we want to train other researchers, right? You need a research coordinator, you need a research nurse, you need somebody who is going to take care of regulation. So we want to enhance those infrastructures. Next slide. So these are the presidential scholars. I will not go to details. If you have any questions, please ask Dr. Palmer to explain that to you. And the Dean Scholars, this is the one thing that we really got excited. I actually talked to a lot of medical students. So the idea is you're going to finish your first 2 years of medical school. After that, we're going to pay you for the full year for you to do research. And then you're going to go back to medical school. And when you talk to the medical students right now, their board examination, the USMLE, used to be the step one is the one that we decide whether we want you for our residency program or not. Right now it's a possible fail. So you really can't tell who's good or bad. So right now there's a strong interest to do research if they want to get into competitive specialties, anesthesiology, dermatology, neurosurgery. So there's a huge need for those. And next slide. And this is the core that's led by Dr. Megan Cohn, the research design, compliance, data management. And as every time we do clinical research, there's a lot of data, there's a lot of regulation, there's a lot of compliance we have to take care of, and most physicians are not trained to do so. And we learn statistics in medical school, but really not that much. For us to perform statistical analysis by ourselves, it's a disaster. So this core, we will provide research design, statistical support, policy compliance, and data management, and really from the get-go. Before you get the clinical trial started, from the project planning, this core will be engaged, will guide you through how to collect the data, how to analyze the data, how to present the data, and then work with the existing EHR, electronic health research database. Next slide. And our community engagement and outreach core is led by Dr. Dine and Dr. Kidd. So the purpose of this core is really to raise awareness of community health research needs and improve access to relevant research study and data. We all know Louisville, we have a lot of issues regarding our DEI efforts. And West Louisville, many patients, they do not have access to healthcare, let alone healthcare research. And there's a strong distrust of those communities to the researchers. They just don't trust us to do research. And so our goal really to improve that relationship and develop and support community-led and clinical investigator-initiated community-based participatory research. So those are kind of the summary of the center. And this center will work very closely with the CIEHS. And a lot of us actually serve dual roles in this CTR-D grant and also the CIEHS. And I think several focuses for at least the initial funding will be environmental health, cardiovascular, pediatric research, and certain other places. Everybody's open to apply for those fundings, but I think the strongest 3 points will be those. Thank you.
Great, thanks, Jiapeng. So just would like to thank Drs. Conklin, Palmer, and Huang for getting through a lot of material and getting done just about on time. So we covered a lot of material that's just listed there. So next slide, I think we got about 5 minutes or so left. So happy to take any questions. I think there have been some that have gone through on the chat. I think a lot of those have been answered.
Matt, I guess I have a question for for Kenneth and Dan and you too. So for those services, is there a standard pricing that listed on the website somewhere so people know how much that will cost and what, like, 100 miles versus 50 miles, So I think, at least for me, I really kind of want to know that before I, want to design my experiments.
So I can only answer on the CPM side, Jiapeng. So from the CPM side, I didn't mention that we recently got an NIH UC7 grant that completely funds our BSL-3, ABSL-3 research. So we do not charge for using the CPM BSL-3, ABSL-3 facilities. We don't charge for use of technician time. The only cost would be from our side, the cost of animals and any kind of research reagents that you might need to use. So the CPM heavily subsidizes infectious disease research from that side.
Thanks.
Yeah, Jiapeng, good question. There's no specific single charge that can be conveyed in a menu. So if you have to look at the menu, you can't afford us, kind of thing. No, I'm kidding, but all inquiries have to go through me through a consult first. And then we can talk about, because if the grant involves anybody on the team, if there's support there, that changes the equation. But usually the charges are nominal. On a daily basis, the most I think we charge is about 220 per day, which would be we do everything. We load your animals, we run the exposure, take them out and put them back in the facility. It's more of the logistics of the discussion because it is a barrier facility. Your mice would have to come into the facility, be ordered into there, or be allowed to be transferred from another barrier facility. And then we'd have to talk about logistics of when you wanted to do so more the consultation is the key than the pricing, I would say.
Got it. Thanks, Dan.
Yeah, I'd like to add just something on that everybody keep in mind that CIEHS does offer financial support for research through the core utilization subsidies, which can go up to $10,000 to offset any costs that you might incur, and then bigger awards such as the pilot project program. All right, any other questions? Okay, so before we go, I'll wrap it up. I'd like to make a call and encourage people to consider doing something like what Dan and Ken are doing now, and looking at environmental health and pathogen interactions. So I think this is a very unique feature of our center's renewal application. There's not too many places where you can do this type of research in the United States, if any. And as Ken mentioned, the changes in mosquitoes and re-emerging infectious diseases. Zika virus was on his list, many others. It would be great to not only study those, but look at interactions or how exposures to things that pollution that we know is going to go up with global warming, particulate matter, whatever, how is that going to affect the pathogen? Is it going to make it more virulent? How is it going to affect the host? How is it going to affect the host response to pathogen? I think this is going to be increasingly important, and nowhere else in the country probably can it be done other than here.
Matt, I think Chris got a question. Chris got his hand raised.
Hey, Chris.
Yeah, it's not so much a question as you might want to mention that the session was recorded and the slides in the recording will be posted, links to them.
Okay. And Jessica just sent something, Colleen, so I listed the email contact as TRSC@louisville.edu. So I don't think we've updated that yet. It's probably still IHSFC@louisville.edu, Integrated Health Science Facility Core.
Yes, that's true.
So Colleen, let's create a new email address since the program NIH made us change the name of the core, and we're still tying up all those loose ends. So thanks, Jessica, for pointing that out. So anybody can email me directly as core director or IHSFC@louisville.edu. Great. Thanks, everybody. And so we'll be back in next quarter, and we'll have one of the other support of the 4 research supported areas. We'll have a presentation on one of those. I can't remember which one, but it'll be GIS or EPI most likely.
Right. It's August 21st, 3:30 to 4:30. Dr. Janice Sullivan will talk about pediatric research.
Okay, pediatric clinical research.
Yep.
All right, great. Thanks, everybody. Thanks, Jiapeng.
Thanks.
Thanks, Matt.
Bye. Thanks, Matt.